Hi everyone, let's talk about protein dysfunction. How does it occur? So, dysfunction is caused by mutation of gene coding for that protein. A gene codes for a protein, and when there is mutation occurring in the gene, the structure of the protein is changed, which leads to change in function of the protein, which causes protein misfolding or protein dysfunction, and hence protein doesn't work. In this case of phenylketonuria, phenylalanine hydroxylase gene, more than 600 mutations have been identified. Most of the mutations are either missense or point. Now let's talk about missense mutations. So missense mutation is one of the point mutations where one amino acid is swapped with another amino acid. So in this case, the base pair C is swapped with base pair A. So here in this sequence, proline is changed to threonine. This is an example of missense mutation. Now frame-shift mutation. So frame-shift mutation means insertion or deletion. Here, these two are deleted. So C shifts and attaches to G. So you can see here, and the reading becomes completely different. So serine, valine, proline, tyrosine, serine, valine, leucine, leucine. So all the amino acids after the point of deletion or point of insertion changes completely, and hence the proteins becomes dysfunctional. Now let's look at some of the examples of common mutations. R111X, which means arginine at position 111 is deleted. So what happens if it is deleted? Let's go to position 111. So it lies in N-terminus region. So if arginine is deleted in 111, then that causes frame shift mutation, which changes the entire sequence of the protein. Hence, N-terminus becomes dysfunctional, and then protein misfolding occurs. So the binding of the so nothing can control the allosteric inhibition or allosteric attachment regulation of the enzyme. So the allosteric regulation of the enzyme is not controlled because the cyanide N-terminus is mutated. Similarly, when mutations occurs at catalytic domain indicated by blue, so let's look at one of the example. R241C, where arginine at position 241, it's a missense mutation. So arginine at position 241 changes to cysteine. So arginine changes to cysteine, and what does it do? So the missense mutation has occurred here. This alters the catalytic domain structure, and hence the active site won't be able to bind to the substrate. So if the substrate is not bound, then the enzyme is not functional, which results in phenylketonuria. That we'll talk about in next web page, like in next page. So let's talk about the mutation in general. Chromosome PAH gene lies in chromosome 12. So the red indicates where PAH gene lies, and that's the locus of PAH gene. It has 13 exons, and most the mutations occur most frequently in exon 6, exon 7, exon 11, and exon 12. So this is where the point mutation occurs. The mutated exon has at least one insertion, deletion, or swapping of amino acid residues. So that's how the mutation occurs. And mutation in any one of these domain alters the function of the domain. So if there's mutation in tetrameric domain, then the protein won't be able to form quaternary structure, and hence there won't be active form of PAH enzyme. So the enzyme is in idle form, not an active form, and hence enzyme doesn't work, and enzyme is dysfunctional. That's all about the mutation. Now, moving on to consequences of mutation, as I have talked earlier, mutation in any of this protein domain alters the structure of protein domain, and if it alters the structure of either one of its domain, so the domain fails to perform its function, which results in which results in protein being not functional. Thank you.